Good afternoon, and welcome to today's webinar, Understanding Fecal Coliforms and E. coli Testing Methods for Wastewater in Minnesota. I'm your moderator, Nancy Leonard from IDEXX Water Microbiology. There will be a question and answer session at the end of the presentation, but feel free to submit your questions at any time during the webinar. You can use the Q&A tool at the bottom right-hand side of your screen and type in your questions to the chat box. Your question will go to the panelists only, so the other participants in the webinar will not be able to see your question. We'll try to answer as many of your questions as possible, but if we run out of time and you still have questions, or if you're watching this as an on-demand viewer, you may submit your questions to water@idexx.com. This webinar is being recorded, and a link with the recording will be emailed to everyone who has registered along with a copy of the slides. This webinar will introduce you to indicator bacteria, review the enzymatic test methods for wastewater, quality control, QA and QC, and discuss specific wastewater regulations for the state of Minnesota. I'm happy to introduce our speakers today. Our North American Regulatory Affairs Specialist, Jody Freimeyer, will be walking you through the specific wastewater regulations for the state of Minnesota. Jody received her undergraduate degree in preclinical naturopathic medicine from Johnson State College in Vermont, and is currently studying at the University of Nevada to earn her master's degree in public health. She's been a part of our water business for 5 years and works with a variety of regulatory agencies to help obtain regulatory approvals for IDEXX methods. She also participates in several standards organizations, including the NELAC Institute and ASTM. Before we get to the regulations that affect your state, please join me in welcoming Sean Dubois. Sean has a BA in biology and a BS in environmental science, with a concentration in marine biology and marine ecology. Sean has been a part of our water business for over 17 years, working in research and development, and in his current role, managing the product support group. Welcome, Sean.
Thank you, Nancy. Thank you, everyone, for joining us today. So, as mentioned, I'm going to go over indicator bacteria and testing for contamination and wastewater testing. So, to start off, we're going to talk about indicator bacteria or organisms, why we look for them and what they are. So, in general, when we think about the contaminations that can come from wastewater or contamination, there are a large variety of pathogens that could be present. And, these pathogens can be very difficult to find, they can be dangerous to handle, and can be expensive and time-consuming. So rather than spending all this time looking for certain things, what we can look for is an indication of the contamination by an organism, a bacteria. So some requirements for an indicator organism, because we're looking for them to show whether or not the pathogens are present. So they should be present when pathogens are present, and they should be absent when pathogens would be present. So if the water is contaminated, they should be there. If the water is not contaminated, they should not be present. We want them to be in high enough numbers. We want to be able to have an organism that's very easy to find in high numbers, easy to detect. They should also be able to survive better in the water than the pathogens, because we want to have something that could be in the environment long enough for us to be able to find it, and therefore we can know that the contamination event occurred. They should be easy and safe to analyze. Once again, we want to make sure that any kind of testing that's being done is safe for people working in the labs. Whereas a lot of pathogens, there could be a lot of safety concerns trying to analyze them. So indicator organisms should be easy and safe to handle. Rapid results. We want an answer, and we want it as quickly as possible so we can act as quickly as possible. And a lot of times pathogens can take a lot more time and a lot more difficulty. So having something that's very quick is very important, inexpensive, and above all, accurate. We want to make sure that when we are testing and we are looking for something, that the results that we're getting are true and correct. So there are different types of indicator organisms that can be looked for when we're talking about wastewater testing. For example, probably one of the best indications of fecal contamination is E. coli. So E. coli is a major inhabitant of the intestinal tract of warm-blooded animals, including humans, of course. They are only found, enterically, they are only found in fecal material. So they don't naturally occur in the environment on their own. So this is a indication of a fecal contamination event. They are found in very high numbers when they are shed from the body. So basically, we have something that is clicking all those boxes of what a good indicator organism. Relatively handled and safe, no issues there. So they grow very quickly. So results from this kind of testing can be done very fast. So we have something that's safe, fast, only found during contamination, and easy to detect. So some other things about E. coli is that they're a gram-negative bacteria and a thermal tolerance. So they can actually survive in a wide range of temperatures, whether it be around body temperature or even elevated temperatures. So it's a very good indicator organism. And there are other indicator organisms, and we will talk about that during the presentation. So E. coli belongs to a group of bacteria commonly referred to as the total coliform group. So total coliforms there are different genuses that belong to the coliform group. Includes Klebsiella, Citrobacter, Enterobacter, Serratia, E. coli. So all of these are total coliforms, and they are defined by certain chemical characteristics as well as that they grow at 35 degrees Celsius body temperature. Within that group, we have another subset that's been referred to as fecal coliforms. Fecal coliforms are believed to be bacteria that are truly from a fecal source. They grow at elevated temperatures at 44.5 degrees Celsius, plus or minus 0.2. And it was believed that incubating samples at this elevated temperature would isolate bacteria that could only come from a fecal source. As we move on, we've discovered that that's actually not true. Well, majority of E. coli, which we know E. coli are only found enterically, they're not found in the environment naturally. Not all E. coli strains are able to survive at this elevated temperature. Conversely, some of the environmental bacteria, coliforms, are able to survive at this elevated temperature. So the bacteria that are growing at this elevated temperature aren't necessarily from a fecal source, and ones that should be growing aren't. So a better term would be thermal tolerant bacteria, bacteria that are able to survive at elevated temperatures. However, when it comes to regulations and everything, and from this point on, what we'll just refer to as fecal coliforms, that's what people are used to. So when we think about looking for these indicator organisms, which would include E. coli, fecal coliforms, enterococci, there are different methods that can be utilized in order to detect them. And as time has gone on, the methods to do that have improved and evolved. So here we have showing that we've made some changes. So starting off, we have multiple tube fermentation, which is time-consuming and has a lot of steps. There's up to 60 steps total in order to run this method, with results, confirmed results, taking up to 4 total days. So you're looking at a very long test, complicated, time-consuming, expensive, but gives you a result. So, it meets some of those checkpoints of some of the tests that we want to use, but not quite the best. As time moves on, we now hit membrane filtration. Membrane filtration reduces the number of steps required to run the test. So we go up to 20 steps to run the method. And confirmed results are between 24 and 48 hours, depending on the test. So a great reduction in the number of steps to get the results and the amount of time. However, it's still relatively labor-intensive. There are plenty of points where errors can occur that would impact results. And it's still a lot of resource and time spent to run this method. And finally, we come to the enzymatic methods, which would include colilert, colilert, and enterococci. So the number of steps to do quantified testing is reduced to 8 steps. And we now have confirmed results between 18 and 24 hours, depending on the test. So a further reduction in the amount of steps required to run the test and the amount of time to get a final result. So to go over colilert and colilert and enterolert, all of these products basically have the same method in how they perform. So these reagents, the powders, contain a variety of ingredients, and they can be kind of grouped into several different categories. You have food for the bacteria, our target bacteria, the stuff that we want to grow and detect, the indicators. Suppressants to prevent other bacteria and other organisms from growing or interfering with the test. And finally, we have we call it the nutrient indicator, the thing that's actually going to tell us whether or not our target bacteria is present in that sample. So when we talk about colilert and colilert 18, when utilizing it for total coliform testing or fecal coliform testing, there are actually two different indicators present in the media. So the first one is O-nitrophenyl beta-D galactopyranoside, or ONPG, and this is the nutrient indicator to indicate whether or not total coliforms or fecal coliforms are present in the sample. So the way this works is that you have a complex sugar, the beta-galactopyranoside, and bound to that is the indicator O-nitrophenyl. When these two are combined with one another, the appearance is colorless. There's no color. Total coliforms have the enzyme beta-galactosidase, which is able to utilize this nutrient indicator. And when that happens, it binds with the sugar, and then the indicator is cleaved, removed off of that sugar, and then will hydrolyze and turn color. And so for OMPG, it goes from colorless to yellow when it's utilized. So this is how we're able to determine whether or not total coliforms are present. So colilert and colilert 18 also have a second nutrient indicator, and this is 4-methylmbifural beta-D glucuronide. So the same concept, you have the nutrient, which is the beta-D glucuronide, and combined with that is the 4-MU indicator. When these are bound together, the sample will not fluoresce when exposed to UV light. E.coli, which is a total coliform, so contains both the enzyme beta-galactosidase, contains a secondary enzyme that's pretty unique to E.coli, beta-glucuronidase. So this enzyme will utilize the MUG substrate, which will cleave with the sugar, bind with the sugar, and then the indicator will be cleaved off. And then once it's hydrolyzed and exposed to UV light, it will fluoresce. So colilert and colilert 18 are able to detect both coliforms and E. coli at the same time. Total coliforms will have yellow color, and E. coli will have both yellow and fluorescence, since it is able to use both indicators. So enterolert works on the same principle. Only difference is that it's a different sugar that's combined with an indicator. So in this case, it's 4-methylmbifural beta-D glucoside. Same thing when the sugar and the indicator 4-MU are bound together. They're not fluorescent, and once it's cleaved off from the sugar, it will fluoresce under UV light. So in this case, enterococci have enzyme glucosidase, and it's able to utilize this indicator. And so each one will provide a signal output, either color or fluorescence, based on the presence of the target organism. So tests are pretty simple to run. They can be run both as presence assays and for quantification. And so the next session we'll go over how to quantify using colilert, colilert 18, and enterolert. So to do that, you need the reagent for your test for your analyte, as well as the quantitray system. And this includes the quantitrays themselves, the quantitray rubber inserts, and the quantitray sealer. Having all of these materials along with the reagent will allow you to quantify your sample that you want to test. So steps are, once again, it's pretty simple. You would take your sample, add the appropriate reagent. In this case, we'll talk about colilert and colilert 18. But you would add the reagent to that sample, shake to dissolve, and once it is completely dissolved. That would then get poured into the quantity tray. The quantity tray is then fitted into the insert, which is then put through the sealer to seal the sample. And basically the way that the tray works, the tray is actually, in a sense, at this point, is like a bag, where you have the front part, which has the plastic and has the well shapes, and the back, which has a foil with an adhesive. And basically what happens is you pour that sample in there, and when you put it through the sealer will dispense the sample into all of the wells, while at the same time will activate the adhesive, which will compartmentalize the sample into all those different wells. And this is what allows us to have a quantification of that sample. For colilert and colilert, when running these tests for total coliform and E. coli testing, it would be incubated at 35 degrees plus or minus half a degree. For colilert, the test is a 24-hour test with results valid up to 28 hours. And for colilert 18, it's an 18-hour test with results valid up to 22. So take your sample, run through the sealer, and then place into your 35-degree incubator. And then if you're reading colilert or colilert 18, take it out at the appropriate time, and then read results. So for total coliform, first you would read the tray looking for yellow color. Yellow color indicates a confirmed positive for total coliforms. If any wells that are yellow are found, the next step would be to take the tray and place it under a 365 nanometer UV light. And now you're looking for any wells that are fluorescing, any yellow wells that are fluorescing. Fluorescing yellow wells are considered a confirmed positive for E. coli. Once again, E. coli is a total coliform, so it's able to utilize both indicators, so it has to be yellow and fluorescent to be considered a confirmed positive for E. coli. Now, if you want to run colilert 18 as a fecal coliform test, slight difference. You still take the sample, add colilert 18 reagent, dissolve, seal in a tray. But instead of using 35 degrees, you'll actually be incubating at 44.5 plus or minus 0.2. And this temperature is extremely important for fecal coliform testing. That range will dictate the result that you get. If you have the sample being incubated at higher than 44.7, then the temperature of the water can be, temperature of the sample will get too hot, which will actually kill off and prevent fecal coliforms from growing, which means that you artificially lower your results for that sample. If you're below the 44.3 degrees Celsius, now the sample is too cool and non-fecal coliform bacteria can grow, which means that you actually be having a higher count. So that temperature range is extremely important to maintain, that 44.5 plus or minus 0.2. So we recommend using a water bath for testing for fecal coliforms because it's able to maintain that tight tolerance, temperature tolerance, a lot more efficiently than a air incubator. So after you've taken the tray, sealed, next thing to do is submerge it directly into the water bath, completely below the surface of the water. You don't want to put the tray in any other kind of bag or other container. It would be directly into the water. The tray is designed so it won't allow any water to enter the tray or any of the sample to leave the tray into the water. So no concerns there. And the tray will be all right, putting it directly into the water bath. Then you want to be able to weigh it down with an appropriate weight to make sure it maintains below the surface of the water. And you can see on the picture here there are a couple of weighted rings. These are great to use. They won't mess with the thermodynamics of the water and allow the sample to come to the correct temperature and maintain that temperature. What you don't want to use is something like a glass bottle filled with water or a brick, rock, anything that could damage the tray or could mess with the thermodynamics of the water and create like hot and cold spots, and that would actually influence the test. So you want to make sure that you use the appropriate weight to maintain the trays below the surface of the water. Once again, this is colilert 18, so it's an 18-hour test with results valid to 22. And then after the 18 to 22 hours, you remove the trays from the water bath and then look for yellow color development. Once again, it's only yellow. Yellow color indicates a confirmed positive for fecal coliforms. Because as discussed earlier, fecal coliforms can be both E. coli and other total coliforms like Klebsiella. So we only need to utilize the yellow indicator. We're not looking for fluorescence. When it comes to colon and collutein, there is a comparator that can be used when reading results. And what the comparator does is it provides a minimal amount of signal intensity in order to call something positive. So it has a minimal amount of yellow and fluorescence to say that the result that you're seeing is a positive. So anything that is equal to or greater than the comparator is considered a positive. So this will come in handy, especially if you have new users or new people to the test. We just need to understand and see what a positive looks like. It's important to have for audits, audit purposes, and just maybe for trainings. So it's a good tool to have. Switching gears to enterolert. So if you're running your sample looking for enterococci, slightly different. You go through the same process of taking the reagent, dissolving it in the sample, sealing it in the tray, and then incubating it. Except that now we're going to incubate it at a different temperature. It's going to be at 41 degrees, plus or minus half a degree Celsius. Time to result is the 24-hour test with results valid up to 28. So after the 24, 28 hours of incubation, remove the tray, and then put the tray under a 365 nanometer UV light. And what we're looking for is blue fluorescence. Blue fluorescence indicates a confirmed positive for enterococci. So at this point, whether you've been running coliform, E. coli testing, fecal coliform, enterococci, you have trays, you've counted the number of wells, now what do you do? Now it's to confirm what your number of for your sample is, you have to calculate the most probable number for that sample. So to do that, you basically count the number of positive wells in your tray, and then refer to the MPN tables that's included with each type of tray. So the Quantitray 51 well has 50 large wells of one type, and then on the top of the tray is one large well, and that's the 51st well. This will give you a counting range of around 200, up to 200. The Quantitray 2000 has two different size wells. It has 48 small and 49 large, with 48 being of one size with one large one at the top, which makes it the 49th well. So what you would do is you would take your tray, count the number of positives, and refer to the MPN table. So for example, a 51 well, if you had 4 positive wells in your sample, you would look at the MPN chart, and this would tell you that your result is an MPN of 4.2 for that sample. Once again, the Quantitray 2000, you have 2 different size wells, so you actually need to count both of them at the same time. So you would count the number of positive large wells, record that number, and count the number of small positive wells and record that number, and then refer to the 2000 MPN table. So in the case of this sample, if you had 48 large positive wells and 25 small positive wells, this would give you an MPN of 344.1 for that sample. So depending on what kind of sample you're running and the expected result, you can choose one tray over the other. But if you always be below 200, then you use Quantitray. If you expect the possibility of having higher counts, then the 2000 will give you a better option, as the results will give you up to 2400. One thing that often comes up, at least since I've been here, and I know it's been on for quite some time, is the difference between the nomenclature of MPN versus CFU, especially for people who are used to running membrane filtration. And so the question is there a difference between MPN or CFU? And the real simple answer is no, there is no difference. The most probable number, MPN, or coliforming units, or CFU, basically refer to the type of test that was run in order to obtain your result. So if you are running a sample using a liquid-based media, such as colilert, the results will be reported out as MPN. If you're running something on a solid media, such as an agar plate, then the result would be a CFU. basically all this number, all this MPN or CFU is just the label associated with the result that you get from your test. Both of them are appropriate to use. Not one is not better than the other. And the other important thing to point out is both of these have a 95% confidence range. Some people believe that a CFU or an auger plate gives you a definitive number, and the truth is, if you look at certain methods and other resources, there is a 95% confidence range built around those tests as well, because there's a lot of variation that can occur. It's not guaranteed that all the bacteria are surviving on the filter. The filtration process is kind of harsh, so you might have some loss there. There's a bunch of other processes. So basically, what it comes down to is that both methods are very similar. The result of MPN versus CFU there really is no difference. It's just saying what kind of testing that was done to get your result. Once again, as talked about with the Quantitrace, one of the big advantages to the Quantitrace system is that it gives you a very large counting range. So you have up to 200 with the Quantitray and up to 2,400 with the Quantitray 2000. So that should meet most people's needs. However, if you have a sample that may have a count that would be outside of the Quantitray's range, dilutions can be done. The important thing to know is that when you're going to dilute your sample, the dilution has to occur before you add the reagent. So if you have a sample that you have concern that might be kind of high, you would do the dilution prior to adding the reagent. So perform the dilution, then you would add your reagent, then go through the normal processes of making sure it's dissolved, add to the Quantitray, seal, and then incubate. Then after you read the results and record your MPN results, the next thing to do is to correct for your dilution factor. So if you did a 1% dilution prior to running the sample, you would take whatever result you got for that sample and multiply it by 10 and come to your final result on a per 100 ml basis. Okay. Switching gears, looking more on the quality control now. So quality control is an important thing to do whenever receiving new lots of material, and you want to make sure that it is performing as intended. So in the case of IDEXX products, usually what's suggested is to run a positive and negative control when each new batch is received. What we like to point out is that depending on what method you're running will actually determine the amount of QC that's required. So here's a simple graph of looking at colilert 18 and the QC steps that are required versus MFC, and this is for fecal coliform testing. Recent method update rule increased the amount of QC that's required in order to run the MFC method. So as mentioned, colilert 18, when a new lot is received in, basically it's suggested to run a positive and negative control to verify that you're detecting the target and not detecting non-target bacteria. And basically that's all you need to do for QC. With MFC, there is the requirement of running positive and negative controls when you receive a new lot, create a new batch. Then there's also on top of that verifications on a per lot basis. You're running multiple blanks during each sample run, and depending on the number of samples that you're running, you will increase the number of blanks that are required. And then on a monthly basis, you have to verify the colonies that you're counting to confirm that they actually are a fecal coliform. So there's a lot more QC required for membrane filtration than there is for the enzymatic colilert 18 method. So when it comes to QC, we would suggest using a strain that comes from a national collection, such as ATCC or NCTC. These can be obtained and maintained. IDEXX offers a QC line of products, IDEXX QC, that are relevant for each of the types of products that can be purchased. So there's a total coliform E.coli kit that will give you E.coli as your positive for E.coli, Klebsiella varicola for total coliform, and Pseudomonas aeruginosa as your non-target. For fecal coliforms, it'll have a fecal coliform for E.coli and thermotolerant bacteria. Pseudomonas aeruginosa as your non-target. And then for enterococci, you get an enterococci E. fecalis as your positive control, and then two negative controls, one being a gram-positive bacteria and another gram-negative bacteria. So these are simple to use and can make the QC process a little easier than having to maintain cultures. But either way is appropriate to QC the product. Continuing on the QC of product. So a lot of QC is done internally before a product is released. And what this will generate is a certificate of analysis that will let you know the testing that was done on the product prior to being released and whether or not it passed. This certificate analysis is important to have for your records, just so you have it with your materials, and also if you have any audits that come through. So you want to make sure you have CFAs for the reagents and media that you're using. For IDEXX, all of our CFAs can be found on our website. And the link is here in the presentation, and I know that Nancy will send this out after everything is done, so you can click on this link. Additionally, just go to our website, IDEXX.com/water, and there are several different ways to get to the CFA from there. Basically, you'll have a search field. You'll type in the lot of the product that you're looking for, and links to PDFs for any product that's associated with that lot number will come up, and you just click on that, and you'll have a PDF that you can download or print. And basically, the CFA is going to tell you all the different testing that was performed on that product in order to confirm that it was tested. So basically what ends up happening is that for our reagents, all the reagents are made here in Westbrook, Maine. The reagents are produced, and during the run of that lot, samples are collected from across the entire lot. Those samples are then given over to our quality control lab, who will run a battery of tests to ensure that the product is performing correctly. Once it's been confirmed that all tests have passed, that will then get reviewed by several different individuals. And once that's all been accounted for and corrected, that will get sent off to our quality assurance team. They will then review the packet and any other associated materials that need to be in for that lot. And only once everything has been completed and shown to be correct, will QA create the certificate of analysis, post that to the site, and release the lot for distribution. So we have several steps in our process to ensure that the product that we're releasing is of a high quality for our customers. IDEXX has a strong commitment for quality. IDEXX, as a whole, has several ISO certifications, 9001 and 14001 for the entire company. And then our water QC lab specifically has ISO 17025 for most of our products. And what this does is it's another certificate saying that we put a lot of it into our testing for our quality control for our products. And for countries or organizations that follow ISO standards, having this certification from our QC lab actually reduces the amount of QC testing that a customer might have to do on their end, especially in Europe. But it just goes to show the amount of time and effort we want to put into the quality of our products, the importance to us. So once again, all of our products in the water business have an associated CFA, and those can all be obtained on our website, whether they be the reagents, the quantity trays, our vessels, the IDEXX QC, all of that can be found on our website. So, thank you.
Thank you, Sean. A lot of really good information in there. I really appreciate your time. And just as a reminder, if you do have questions for Sean, you can submit them at any time before we get into Joey's portion of the presentation. Just type in your question in the chat box in the Q&A tool at the bottom right-hand side of your screen, and we'll get to those after Jody's presentation. So now we turn it over to Jody. Jody?
Thank you, Nancy. Hello, everyone. So the U.S. EPA drives regulations in Minnesota, just like it does for all other states, and as a way to control the discharge of water pollutants to surface waters, the Clean Water Act was implemented, and these regulations can be found in the Code of Federal Regulations at 40 CFR Part 136. Every so often, the EPA updates these regulations through a process called the Method Update Rule. And the Method Update Rule completed in August of 2017 included the EPA's final approval for IDEXX's Colilert 18 fecal coliform method for wastewater effluent testing. After the method update in 2017, states that have primacy can either incorporate the federal rules into the state rules, or the state can create rules that are equivalent to the federal rule. But if the state does create its own rule, it must be as stringent as the federal rule. It can't be less stringent. And just to point out, there are a few hyperlinks within some of the regulatory slides. So once the presentation is available, if you'd like to review any of the regulations or contact links, you can just click on the hyperlinked word that's underlined in blue. Wastewater effluent monitoring is enforced through the National Pollutant Discharge Elimination System Permit Program, which is referenced as NPDES for short. Wastewater bacteria parameters include fecal coliforms, which are included within the Minnesota Administrative Rules under Part 7001.1060. And testing done for NPDES fecal coliform requirements must be done with an analytical test method that is included within the U.S. EPA CFR Part 136, specifically methods that are included under Part 136.3, which with the EPA's 2017 method update rule, colilert 18 fecal coliform test is now included within that section. But it's also important to keep in mind that it's possible that some wastewater permits could list a specific test method for bacteria requirements instead of stating the general allowance to use any EPA-approved test method. So if there were questions on the way an NPDES permit is written, or if the permit lists a specific test method, it's best to reach out to the permit writer listed on the individual permit, or you could reach out to the appropriate regional NPDES permitting office. So since the EPA has adopted the method update rule, which included adding the colilert 18 method for fecal coliform testing, this change affects wastewater laboratories in Minnesota by allowing more analytical test methods that can be used for NPDES permit fecal coliform monitoring requirements. And this revision can be really helpful to laboratories since, as previously mentioned by Sean, colilert 18 reduces quality control steps, reduces hands-on time, makes demonstration of capability easier for both a new analyst or for any ongoing DOCs that might need to be continuously done over time, and it allows for more reliable data. So in the state of Minnesota, any microanalysis completed for NPDES permits must be done by an accredited laboratory. And facilities that have a Minnesota Pollution Control Agency issued NPDES permit, such as a municipal, government, or industrial site that performs in-house testing, are required to be accredited through the Minnesota Pollution Control Agency. All other wastewater testing laboratories, such as the commercial laboratory, are required to be accredited through the Minnesota Department of Health Environmental Laboratory Accreditation Program. But even though there are two separate programs, the accreditation requirements are very similar, and the overall process of adding a new method applies to both programs. However, accredited laboratories will need to follow the appropriate requirements for changing or modifying their current scope of accreditation, but the overall steps include completing the applicable forms and paying the associated fees. And just to point out a small difference here between programs, the Minnesota Pollution Control Agency Laboratory Certification Program provides the application form right on the website, but for the Minnesota ELAP, laboratories would need to reach out to the program to request an application. Other steps include reviewing and possibly updating the Laboratory Quality Assurance Manual, writing a standard operating procedure for the manual, which as part of the SOP, you'll want to include the quality control requirements, which are listed within the colilert 18 package insert. But if you did feel like you needed some help with creating an SOP, here at IDEXX, we've created an example SOP document that is available by request. Also, laboratories would need to complete and document the initial demonstration of capability, participate and pass 1 to 2 proficiency tests, and potentially complete an on-site assessment. Now, just to point out another difference here within the PT requirements for the Minnesota Pollution Control Agency Laboratory Certification Program, labs will need to successfully complete at least 1 proficiency test, but the Minnesota ELAP program would require passing 2 proficiency tests. Also, just to mention, I put together a document that outlines today's regulatory content in the form of a method adoption guide that is specific to the state of Minnesota, and it includes the steps for changing an accreditation scope. And this informational guide also includes the example SOP document, and if anyone would like a copy of this guide, please feel free to reach out to us and request one. So to wrap up today's regulatory portion, I'd like to share a part of what I do, which is I follow various state rules and regulations to see how they periodically change over time. And it's fairly easy for anyone to follow and even participate in Minnesota rule changes because anyone from the public can comment on proposed rule changes, and comments can be quite impactful to rulemakers, especially when they come from stakeholders such as wastewater utilities or laboratories, since you're the ones who do this type of work and you might be directly affected by any rule changes. A comment submitted to the state can include things like your opinion on the rule subject, any data you'd like to share that might be impactful pertaining to the regulation you're commenting on, or how the proposed rule change may impact your daily work. And you can sign up to receive email notifications on various rulemaking activities so you can watch and track what's happening in your state. And that concludes what I have for the regulatory portion today. Thank you.
Great. Thank you so much, Jody. We really appreciate your time as well. If you do have any questions for either Sean or Jody, again, you can just type in your question in the Q&A tool in the right-hand side of your screen. And we do have a couple of questions that have come in. First question is for you, Sean. For fecal testing, do you use the same Quantification 2000 using colilert 18, water bath for 18 to 22 hours? Did I get that right?
You got that right. It's the same tray for all of the products. It doesn't matter on the product. So yes, it would be the same Quantification 2000 used for any of the tests. Yep.
But the incubation temperature?
Oh, yes, incubation, yes. So incubation is correct, 18 to 22 hours, and a water bath being the optimal way of doing that. Yep.
Okay, great. Thank you. This one is for you, Jody. Follow up to also again to doing fecals with colilert 18. Do commercial labs need to add colilert 18 fecal coliforms to their accreditation scope?
Yes. Yes, they would need to add a new method to the lab scope of accreditation. Yeah.
Okay. And how would they do that?
Well, we do have the method adoption guide that we can certainly send out. If that person would like a copy, we can go ahead and send it out to them.
All right, great. So as Jody had mentioned, she did put together a method adoption guide specifically for the state of Minnesota. And if you would like to receive that guide, you can send us an email at water@idex.com, or you can respond to the email that you'll get from me in the next couple days. That will include the presentation, a PDF of this presentation, as well as a link to the recorded webinar. This one is for you, Sean. Is it acceptable to make a dilution of a wastewater sample?
Yep. Yes. So if you were assuming, if you had any kind of past history knowing that the sample could be outside the counting range of the quantity tray, as mentioned before, you can do a dilution prior to the sample. And if there is any concern about the appearance of the sample having any kind of color or turbidity or anything like that that might interfere with the read of the test, then you can most definitely do a dilution for it.
Okay, so you would do that if there is turbidity or sediment in the sample as well?
Yep. Yep. Anything that would interfere with it, a dilution would be acceptable, and it will help with the read quality of the test. Yep. Okay,
great. Thank you. Another one for you, Sean. A lot of questions about fecal coliform testing. Is there a different colilert 18 reagent for fecal coliform testing versus E. coli testing?
No, the same reagent is used for either E. coli testing or fecal. What differentiates the two tests is the incubation temperature. So it's the same thing with the quantity tray, it's the same quantity tray, everything is the same. The only difference is how you run the test. So if you're doing it for total coliforms and E. coli, you would use the same reagent, the same tray at 35 degrees. But if you're doing it for fecal coliform, it'd be the same product, you would just do it at 44.5.
Okay, great, thank you. And is it okay to use an air incubator rather than a water bath?
We suggest using a water bath as the best way to do it. It's able to maintain the tight tolerance. Again, that's really important. So a water bath is able to maintain that plus or minus 0.2 a lot better than most air incubators. There are some really high-end air incubators that might be able to do it, but we would recommend using a water bath.
Okay, great. Thank you. This one is for you, Jody. What do I do if my permit states a specific method?
Yeah, so if a NPDES permit lists a specific method, then you would want to contact the permit writer listed on the permit, and then you can ask what needs to be done to have the method changed.
All right, great. Thank you. A couple more questions have come in. Can I use colilert for fecal coliforms? I guess that means they're asking if they can use colilert instead of colilert 18.
Nope. Only colilert 18 is approved for fecal coliform testing, so only colilert 18.
All right, great. Thank you. And this is a question that I'm also curious about. Why are there smaller wells at the bottom of the quantity tray?
So the smaller wells and the Quantification 2000 specifically are there, and it basically kind of creates a dilution within the actual test. And those smaller wells, having the two different size wells, is what allows us to have a larger counting range in that same sample. So having that 100 mLs separated over two different well volumes allows us to have a counting range of over 2,400.
All right, great. Thanks. And this is the last question for you, Sean. Is there a separate comparator for fecal coliform testing versus just coliform and E.coli testing?
There is not a separate comparator. They are the same comparator. So the only difference is that you're just looking for yellow fluorescence if you're doing E.coli, where you'd only use it for the yellow for fecal coliform testing, is just looking for that yellow color development.
All right.
So one comparator.
Just one comparator. Okay, great. Thank you. So we appreciate your time today. Appreciate your time, Jody, and yours as well, Sean. And for you out there listening, we know that you are all busy as well, so thank you for joining us this afternoon. We hope that this was valuable time spent for you. But before we let you go, we just wanted to let you know about something that we think is really exciting. We just launched the IDEXX Water Academy. It's an online learning portal where we have e-learning modules, videos, protocol information, and we even have our archived webinars on there. When I send out the link with all of the other information, either this afternoon or tomorrow, I will also be sending some more information about the Water Academy. As I said, we just launched it, so we'd love for you guys to take a look and give us some feedback and let us know what you think. If you can't wait for my email, you are more than welcome to go to idexwateracademy.com and check it out before I send you the link. So we appreciate your time, and as always, if you do have any questions for us, or if you're watching this as an on-demand viewer, please send your questions to water@idexx.com. Thank you again for joining us, and we hope that you enjoy the rest of your day. Thank you.